안녕하세요. Good morning, everyone. Thanks for attending the webinar today. I'm Nicole, the regional business manager from Azure Biosystems. Today, we will conduct the webinar in English together with our local current distributor, Philip Korea. So I would like to introduce the speaker today, who is Dr. Tan Kai-Ling, the regional field application scientist from Azure Biosystems. Kai-Ling, please take away the webinar today. Thank you.
Thank you, Nicole. Hello, everyone. Just before I start, I want to check if my voice is clear for you. And can you see the slide that I display on your screen? If everything works fine, please type yes in the chat box. Great. Thank you, Nicole. So let us start the webinar. Today's webinar will be on the topic beyond the fluorescence glass slides, and we will focusing on the flexibility of imaging and the scanning technologies for high impact and scientific research. The product that we will be using it as an example will be the Sapphire biomolecular imager that we have in our company portfolio. So I'm Kai-Ling, the Regional Application Scientist of Azure Biosystems. So let me start with a very brief introduction to our company. We're a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. We produce and manufacture products that combine smart and simple workflows with high performance and affordability, so you can have the confidence in your data to move forward quickly and the flexible capabilities to go whenever the data takes you. Our company founded in United States, is based in United States, in near San Francisco, very near the Silicon Valley as well. So here's some leadership information on our company. The company is founded by Arnold Shifji. He is our CEO and chairman. He is a venture investor, and also Dr. Diping Che. He is our executive vice president of R&D. If you're looking back in his past experience, he has a wide variety of experience in developing protein imaging system back in Alpha Innotech and Illumina. Here's some simple timeline of our company portfolio. And our company is a fairly new company, founded back in 2013. All the core members are coming from Alpha Innotech. And in this year, 2022, we have 3 key instruments in our company portfolio. The first one, and also the stars of today, is Sapphire biomolecular imager. This is the industry first hybrid CCD imager and laser scanning system. And of course, we have Azure imaging system that is focusing on Western blot imaging, and Azure also that cover both Chemi and Fluorescence Western blot imaging. And we also have qPCR system as well, where we launched in 2020. So basically, Azure Biosystems, we are a Western blot solution provider. We have gel systems, reagents, and consumable for the user to perform Western blot, and also the imaging systems that come along with it to take the Western blot images. And today, we will be focusing on this piece of instrument, which is Sapphire biomolecular imager. Here's our agenda for today. Of course, I will introduce the excitation and detection technologies that is in the Sapphire biomolecular imager that allows the user to do RGB near-infrared fluorescence imaging, Chemiluminescence imaging, and also Phosphor imaging in a single machine. Then next, I will extend the application to a Western blot, in particular Fluorescence Western blot, that will give you a better quantitation power if you are doing a Western blot in your studies. Of course, lastly, we will look at those amazing research applications and also publications that has been done using the machine. Right, so this is Sapphire Biomolecular Imager. It is the first and only laser scanner for all the detection methodologies, like say covered, RGB, visible, fluorescence laser detection, near-infrared fluorescence laser detection, CCD-based chemiluminescence detection, phosphor imaging, and visible imaging as well. Right, so the title today is Beyond the Fluorescence Glass Slide. So this also is Sapphire itself. It is a glass slide imaging system. It's a laser scanning system. So there's no doubt that it can scan a glass slide that's giving the fluorescent signals, such as your IHC or ICC slides. Here we have examples from our users. There's a brain slices and also the embryo slices as well. They're stained with fluorescence, fluorophores. So you can actually see their expression and localization throughout the area. So this piece of system, or say the Sapphire itself, is meant to speed up biomarker identification workflow in such that we have to use multiple scientific tools such as Western blotting, immunohistochemistry, and even microscope to actually validate the data and do subsequent functional analysis or knockdown or knockout or knock-in. So that's in order to prove the hypothesis at a lot of levels, such as subcellular levels, tissue level, and up to organism levels. And this system is meant to serve that, to accelerate this process, or we'll say marker validation process. So not only that it can do the fluorescence slides that we usually see on IHC or ICC, it can also do chemiluminescence, okay, particularly chemiluminescence Western blot imaging with color marker, and also phosphor imaging, and also any other samples that emit fluorescent signals is not restricted by the means of glass slide, even though it is a laser scanning system. You can use the machine for gel, array, okay, for 96 or 384, organism, animal organs, and even plates as well. Right, so the reason why this piece of machine is able to do that, because the excitation and detection technology within the machine itself. Let's just talk about the excitation. So it's a fluorescence machine, a laser scanning machine. So what it has up to 4 solid-state diode laser excitation light sources. All for 4 different channels. We have RGB, okay, also both near-infrared channels as well. And then with that, we also have combination of 3 different detector, PMT, photon multiplier tube, and APD, avalanche photodiode. These 2 are very good for uncovering both visible and near-infrared fluorescence imaging and also phosphor imaging. Of course, let's not forget about CCD camera, which is still the ideal detector for chemiluminescence blot. Right, so with that, it gives us the flexibility in imaging whatever the sample you have in your lab or in your research. Right, so this piece of machine, if you notice, I have mentioned about solid-state diode laser. So why we have chosen laser in this machine instead of any other type of light sources? Because this machine is meant for higher signal to noise ratio at a better sensitivity and giving you a very clean background. Laser diode can give you that such kind of power, because the laser diode is giving you the monochromatic light source. It is higher energy output, definitely. It's a collimated light as compared to LED, which is a diffuse light. This will give you more energy to the sample, and of course, the sample itself will be receiving the energy and emitting the fluorescent signals much at a better efficiency. In that, how it translates into data will be a very good signal to noise ratio, very clean background, and the signal can be will be way higher and are not easily being affected by the background noises. So that's the main differences between LED and also laser diode. And also one thing is LED, even though it's optimal, we'll say optimal because of the price range and everything, it is still having risk of giving you this, say light leakage. So light leakage, this is the reason why you have background noises in your fluorescence slide or any kind of samples. But laser will not have such problem because they are specific. They are within that wavelength. They are highly energized, and then therefore they will not have this kind of very low or none signal crosstalk occur to other channels. Right, so here's the excitation light sources it has in the machine. As you see, we cover both RGB, red, green, blue channels, and also the near infrared channels. So because you see, we have few options over here for the near infrared or the red channels for the user to choose. These are basically compatible with majority dyes on the market for fluorescence signals, the RGB and 2 near infrared channels. So there's no worries about the dye that you choose. And of course, the combination of these detectors in the system, as a photon multiplier tube and avalanche photodiode, these two are basically more sensitive in terms of detecting fluorescent signals because they can amplify the electrons or the signal greatly with their own vacuum tube. As compared to CCD, is very good in giving you a single field of view image, which is applicable on Western blot imaging. Okay, right. So the reason why it has both PMT and FPD in the system, because as you can see in this quantum efficiency graph, was adopted from very many years ago when they started with this technology, PMT and FPD. You can see the range that is best performed at. So for PMT, the best performance will be, or they have higher quantum efficiency over visible spectra such as Psi2 and also phosphor imaging. And for avalanche photodiode, APD is very good at Psi3, Psi5, and near infrared channel detection. Therefore, the combination of these two detectors will give you the best for both in detecting a whole range of fluorescence signals covering visible Psi2, Psi3, Psi5, and near infrared detection. And not only that, you can also detect phosphor or radioactive samples signal as well. Therefore, the combination power of the detector, like I said, it can cover basically everything from the phosphor imaging, RGB, plus near infrared, and also it can incorporate a CCD camera inside the machine, allowing you to do ECL or chemiluminescence signal detection, either on your blot or array. Laser scanner technology. So this piece of machine utilizes a scan head. It contains a point source laser diode. So you have the laser diode coupling with the PMT and FPD detector together. So the scan head moves back and forth across a defined field of view, exciting the phosphors and also detecting the signal at the same time. So the pixel by pixel. So this is a scan head style scanner laser scanning system. This kind of technology is very good in detecting, if you want to achieve phosphorescent imaging at a higher sensitivity, means a better background or higher signal to noise ratio, or to a very tiny samples you want to look for. And also, it is very good for quantitative Western blot. This is what I mean for quantitative Western blot is fluorescence Western blot. Okay, so it covers very wide dynamic range for that in terms of fluorescent Western blot. And this piece of machine also allowing you to do high resolution scanning up to 10 micron resolution. So this 10 micron resolution is super good if you want to detect fluorescence or any other phosphor imaging signals on tissue sections and arrays. And also not to forget, and cell culture plate imaging as well. But of course, this kind of laser scanning system, it has its own limitation as well. Of course, because due to the scan head kind of technology, it is limited or is not very good in detecting 3D objects due to the limitation in terms of penetration of the signal. And of course, the scanning will take longer. It is not like your camera. You just snap and you get an image. This is point-to-point scanning and point-to-point detection. Of course, it takes longer time. And also, with a higher resolution, it's going to take a longer time as well. And of course, it doesn't have that exposure time kind of concept in fluorescence imaging in this scanning system. It's more or less like you determine the intensity and then intensity of the scanner, then you get the result. Right. So this piece of machine or Sapphire biomolecular imager is a flatbed scanner, if you can see over here. It has 25 × 25 cm field of view for fluorescence and phosphor imaging. So you can arrange your plates throughout the entire flatbed and then scan them one shot or even glass light. And also we have 16 cm × 13 cm at field of view for Chemi and visible imaging. And of course, this piece of machine is allowing you to do manual focal plane setting. Means you can set your focus manually in the system. So it's not a fixed one. You can set it manually according to your sample because we know that different slides or different plates, they have different height. Therefore, you can assure that you will achieve that crisp, nice image by adjusting the focal plane. So as I mentioned before, it can reach 10 micron maximum resolution. Here, so the differences between 100 micron and 10 micron resolution, this is very clearly here. Even though you see that it's a bit blurry, but in reality, this is what we see if you zoom out. What we do for 10 micron resolution is we have this full moon biocalibration slide where the spot center to center distance is 350 microns. So this being a gold standard to check the resolution of a machine. So this machine is capable of 10 micron resolution consistently, and therefore it has been utilized throughout from our data from our user for microarray and any other scanning. The speed of scanning is 50 cm per second, and it's four laser scanning simultaneously. But you could also have the option to scan sequentially, means you want to scan red channel first, then you want to scan the green channel later. That you can have the option to do so. The speed of scanning is still the highest or the fastest on the market, and therefore you can see that if you have a blot or some kind of samples, 25 × 10 cm, it will only take 50 seconds to scan it in 1,000 micron resolution, but for 100 micron resolution, it can only take up to 8 minutes plus. Right, next, let us look at the flexibility in terms of the application. Right, so not only it can do fluorescence imaging on the glass slides, that is something for sure the system can do. So what we can use with this piece of machine is we have no limitation in terms of samples. So like I said, you can scan your blot, fluorescent blot, you can scan your tissue. We're talking about organs here and also plates. With that, let me extend the entire content to Fluorescence Western blot. Right, so I think back in the year of 2014, there has been an increasing concern in terms of the rigor and reproducibility for scientific research and biomedical science binding. Because we do see that in terms of the Western blot, we've been adopting from a colorimetric to Chemiluminescence Western blot, and now we have a lot of concerns in the scientific community, of course, about the consistencies in the data, and of course, the scientific misconduct, and so on and so forth. So with that, NIH has been, I'll say, they come up with a campaign that promote the rigor and reproducibility from the aspect of the journal, okay, the publisher, how they could actually regulate this kind of control and also data integrity in terms of Western blot image. So allow me to explain a little bit about quantitative Western blot. For a measurement to be quantitative, it must meet the following criteria. If you can see here, we have 3 criteria. The first one is it must be generated by a defined process. Over here would be a Western blot technique. And this process generates a reproducible outcome. The outcome must be reproducible and then consistent, which is precise. Then this measurement or the data must reflect the true outcome means it stay true to your sample, it stay true throughout your entire experiment or your study, so which is accurate. So a quantitative Western blot process needs to be well-defined process, and the step followed as consistently as possible. The data needs to be reproducible as well. That is the first thing you need to do. And the data should be consistent with the data collected using an orthogonal method such as mass spec and HPLC, so and so forth. So you must have this consistency between the data from your sample on the same sample that tested for across a different type of techniques. So that's a consistency and also reproducibility features over here. Here's a quote, I'll say a sentence that I adopt from a Jane's publication back in 2015, where the author talking about or focusing on analyzing the critical factors for quantitative immunoblotting. So the author did mention here, just because we can put a number on an image, well, here we're talking about bands, the band signal. It doesn't mean that we should, because it's only allow us to do so if the quantifiable biomolecule, let's say the protein target, is directly related to the quantity that we want to see in the sample. So the signal to the quantity amount must be in a direct relationship. So the higher intensity it is, you have higher signal, the higher protein amount. So that is the direct relationship. Therefore, if this such relationship has been established, the connection where it will be meaningful if we use the technique correctly. Then we can say, oh, in this, through this technique, the protein A is having, let's say, a higher expression than protein B in such control or such group or treatment. So in order to do that, there is a lot of factors in contributing the quantitative Western blot. So one of the, I'll say, key differences that contribute to this, or call it quantitation power, is the choice between the antibody detection in your Western blot, such as chemiluminescence and fluorescence. These are the 2 major antibody detection nowadays. So chemiluminescence itself, as we know, is a light signal generated from a chemical reaction. So you have your HRP in the presence of luminol, so it generates light as a side product. And of course, this is an enzymatic reaction. It's very good in amplifying the signal, but it also affected by the time of your incubation, the ambient temperature, and also the amount or the dynamic range of your ECL substrate, or the half-life of your ECL substrate, and so on and so forth. So if you can see, if you plot it, if you have a high protein abundance, and then you plot it across the exposure time, you will notice that the signal and also doesn't really reflect the amount of the protein across time. And then this is what we call as dynamic rate of reaction, and it's not consistent. Means it's depending on how long you expose the blot for, and how long you incubate the blot with ECL substrate. The result may vary, and then it may vary from season to season as well. So some of you might notice this in your chemiluminescent Western blot, and therefore, that is fact that chemiluminescence is semi-quantitative at best. But this kind of limitation, we won't see it in fluorescence Western blot or any kind of fluorescence application, because it is not an enzymatic amplification of the signal. It is very stable, and then the most importantly, the signal that you get is proportional to the amount that you see in the sample. It's proportional. It's a direct relationship. And because there is nothing that would change the intensity of the fluorescence signal itself, there's only two things that would change it. The first will be the energy output from a laser, and also the second thing will be the amount of fluorophores on the sample. So since on the system, the laser energy will be a constant factor, so therefore it's not going to change. The only thing that you see in your sample, it will reflect the true amount of your protein target. Therefore, it's a static rate of light generation. And all in all, it is a much accurate and quantitative detection for Western blot using the fluorescence antibody detection method. And it also fulfills your publication requirement, because when you image a whole blot, you don't need to cut the blot into a few strips. You don't need to do strip and reprobe. So every time when you capture the blot images, you will automatically capture the entire blot. There won't be any problem if the publisher or the reviewers asking you to submit a whole blot image to verify that let's say your representative image or your Western blot image you submit in the manuscripts are true or not. Okay, so we're talking about data integrity here. So it's very good in terms of that. And of course, not to forget, if we have amateur or junior postgrad, who they not really familiar with this kind of practices, you may actually having difficulty to find this whole blot image after many years when you want to publish the data. And also with these fluorescence Western blot, you can do multiplexing. As we say, each of these protein targets is corresponded by every single fluorophore, as long as they don't have crosstalk. Therefore, you can actually examine the loading control, such as your housekeeping or total protein, and your protein of interest on the same image. So you're comparing bands on the same blot, so there won't be any blot to blot variation much. And also the possibility of losing the protein due to strip and reprobe process. And of course, more than often when researchers or users opt for fluorescence Western blot, they will adopt total protein normalization instantly, because it's improved the signal linearity and the normalization instantly. So they don't need to worry about the biological and technical variation. All in all, fluorescence Western blot is having a higher quantitation power for sure, improves your reproducibility, and of course, is accuracy as well. Right. So another thing is, this is under the context of when you do your fluorescence Western blot and you image with a CCD camera. Next is, if you want to aim for a higher fluorescence Western blot quantitation power, you can do so. How is you having an even better light source, excitation light source, and also a better detector with higher sensitivity. So only that, if we therefore in the Sapphire biomolecular imager, we have been using it for fluorescence Western blot imaging, you can see that the background is very clean, as it uses the laser excitation light sources compared to LED, and it also uses PMT and FPD for fluorescence signal detection instead of CCD camera. Of course, it takes longer time, that's for sure. And another thing, one of the advantages of laser compared with LED is, of course, in the terms of crosstalk. Here we have a Blot, actually from our user. They incubated with IR680 secondary plus their primary antibody. This is the same. These are the same Blot. There is nothing changed over here. It's one single Blot, but the imaging using two different instruments. One is our instrument, which is using the infrared laser plus a PMT, of course. And also you have another machine that has LED excitation light sources, and also the PMT. But you see that the changes is that you will see a significantly lower crosstalk that is leaking into the 800 channels. And then if it does, it will happen so in the LED. Okay, as you see over here, this is what we call a signal leakage. And this will be a problem if you're utilizing near infrared 700 and 800 channel for detection. You wouldn't sure if this signal belongs to your 800 target, near infrared 800 targets. So therefore, if you're doing, like, let's say for fluorescent protein detection, where you're utilizing infrared 700, 800 channels, we highly recommend you to look for laser excitation light sources to avoid the potential signal crosstalk in your sample, in your blot, in your Western blot, let's say. Right. So all in all, multiplex fluorescence allowing you to detect multiple proteins and then normalize them at the same time in a single blot. Improves reproducibility. As I say, comparing bands on the same blot, you can do with housekeeping as well. You can do it with total protein as well. So there's no issue with that. So if you can see here, these are simultaneous detection of our total protein stain called Azure Red with 3 other proteins. These are housekeeping proteins, tubulin, beta-18, and GAPDH, using all 4 lasers on the Sapphire Biomolecular imager. So it gives you such a clean background, so it won't be an issue when you try to analyze the data. Okay, so the power of 4 lasers here is equivalent to a better quantitation power. Not only you can do it on the fluorescence blot, you can also do something, a term that we are more familiar today, which is plate-based assay, or some of you might know it as in-cell Western. Okay, next, let us move on to look at the amazing research application and publication. So all these images that you're seeing on the screens are scanned using Sapphire Biomolecular imager. These are actually the data that we collected from our user with their permission. This, of course, we have publication in fluorescence blot. Okay, you can see the display in their own channels. Okay, in such a clean background. And then we have is a northern blot as well. So you can also display your data in the grayscale. The same thing, which is a fluorescent blot, and page, which is a gel, fluorescence gel. And also, this is another 5' FEM specific lamp product that's stained in this agar gel that image under 488 laser excitation light sources. And this is something we are more familiar with, high resolution fluorescence plate-based assay, or some of you may call it in-cell Western. This is from our user, and we have it in Singapore. We have this in the 96 well, and scan is the 700 channels, and this is scanned in 800 channel, and this is just merely 150 micron resolution. Like you can see clearly the cells and the signals that represents for in each of these well. Of course, people has been testing it at 10 micron resolution for 384 wells. This is another example for high throughput scanning. And these are FIT-C, Alexa Flow 550, red stain, and also near infrared channels as well at 10 micron resolutions. And if you zoom in and look at this 10 micron resolution HeLa cell that's stained with 800 channels of dye, you can see they are staining perfectly nice with each of these cells And then, of course, this is achievable if you titrate the cell numbers accurately. So it won't be just a patch of cell. You can see individual cells that display the fluorescent signals. And of course, high resolution, this is a microarray. Sorry, the slide is wrong. So it's a microarray slide at 10 micron resolution, where people have been using it for their microarray analysis. And looking at the bigger zoom in format at 20 micron resolution. So giving you a very clean background over here. And of course, we scan at 2-4 laser excitation and scanning the autofluorescent signals from the liver. That is the publication that utilize autofluorescent signals from the liver organ. And of course, spheroid, that is at 10 micron resolution, where our user use it to flag the presence or absence of a certain formation in their spheroid. And we have this lymphatic antigen tracking in mouse hind paw. As it is a flatbed scanner, it can fit up to 2 cm height. So anything that is within the height, you can fit it on the system and scan it. And therefore, you can see the hind paw over here. You can see zebrafish that scan that is infected by viral infection and without the viral infection as well. And of course, embryo and placenta, barrier permeability studies, Drosophila phenotype studies, and xenopus oocytes. You can see they put on the glass slide. We can look at the fluorescence label. And of course, we have one from Taiwan that the user or the researchers that injected a 4A8 labeled honey into the mosquito, and you can see that the distribution of the fluorescence signal throughout the mosquito itself. These are scanned at 50 micro resolution. And of course, we have users that utilize this machine to do whole mouse scan, and their neuroscience research. They, out of curious, they scan a whole mouse over here. You can see the distribution of the transgenic mouse. What is that? The fluorescent signals across the first and organ, and also a general localization in the brain's regions as well. iDisco cleared neonatal mouse skull, and also a 2D reconstruction to switch between channels for multiple signals and images, so you can see the visualizing perfectly. And of course, we have another one just for show, not really for show, but it's trying to showcase how what the machine can do with such technologies. Is we scan it at Grasshopper with using D stacking at 800 channels, 10 micron resolution. What you get is a GIF or something like this. You can see the signals, the fluorescent signals across the body. Of course, in terms of biological research, our user has been using it to detect a signal from spinal cord, phosphor imaging, more phosphor imaging on MSAR, and also to do viral RNA decay study, and also the basic chemiluminescence imaging on your Chemiluminescence Western blot, and also chemiluminescent protein array. And also, this publication comes from South Korea, where the users using Sapphire Biomolecular imager to develop their lateral flow immunoassay for the SARS-CoV-2 virus. And if you're interested, we have application notes that is having details on all these applications. You can feel free to download them from our website, or we can request from Philly Korea. And this is a machine, a Sapphire Biomolecular imager, so it can do it has up to 4 laser excitation light sources. Of course, it has each model that fits for your need. So for example, if you want to do near infrared, of course, we have near infrared models for you, and RGB only or full range or only phosphor imaging. And of course, we have optional modules for you to put inside the machine, so you can have the all-in-one machine that you want that fits in your studies or your research needs. There's some selected customer from Europe. We have US and Europe, which is NIH, Merck, Harvard, and so forth. And also for Asia, we have from Sapphire. We have from Duke NUS and also National University and Taiwan, National Universities, and also and so forth. Shanghai Institute for Biological Research, many and many more. And if you're interested, you can go to the Select Science website, which is our partner website. There's a website to allow researcher or user to review their scientific instrument, so you can have a look at how people can address about this, comment about this piece of machine, and what they have achieved with that. And here we are, Azure Biosystems. We have imaging solutions for labs and application. We have Blot reagent as well, OKAO absorbers, microplate reader, Azure imaging systems for your basic gel imaging and Chemi and Fluorescence Western imaging, and also the Sapphire Biomolecular imager, which is still our highest spec in our company portfolio since 2017 until today. And thank you everyone for joining and your attention. I'm Kelly, Regional Field Application Scientist. I will open the floor for Q&A session. Thank you.